I want you to imagine something really simple. You're opening a package, the scissors slip, and you get a paper cut.
Yeah, happens all the time.
Right, nothing major, just a tiny stinging slice on your finger. But what actually happens inside your skin in the exact minutes and hours following that injury?
Most people probably picture a frenzy, right?
Exactly. You picture this immediate frenzy of cellular activity, like thousands of cells instantly multiplying, just scrambling to bridge the gap and seal the wound. But what if I told you that the very first thing your body does is intentionally age the cells right at the edge of that cut?
It sounds completely counterintuitive.
It really does. Today, we're exploring this deeply fascinating paradox hidden right inside your own skin. We're doing a deep dive into a stack of source material, mainly centered around this really groundbreaking paper from Nature Cell Biology. And it proves that cellular aging isn't always the slow, decades-long decay that we usually think of.
Right, because in a fresh wound, it's essentially a 90-minute biological speed run.
A 90-minute speed run. I mean, okay, let's unpack this, because that just challenges basic biology.
It fundamentally rewrites a core rule of cellular biology, honestly. Sure. When we discuss cellular senescence, which is the scientific term for a cell that has permanently stopped dividing and entered a state of biological aging well, the classical textbook understanding is that it is a painfully slow process.
Like years and years, right?
Exactly, or at least days and weeks for a single cell to transition. We usually associate it with chronic stress or disease or just the gradual wear and tear of getting older.
Right.
So to claim that full-blown, undeniable cellular senescence happens in the skin within just 90 minutes of an injury, I mean, that is a massive paradigm shift.
Wait, 90 minutes? That's what, an hour and a half?
Yep, hour and a half.
If senescence is basically the slow accumulation of damage, how can they be sure? Like, if a researcher looks at a wound 90 minutes after a cut and sees cells that aren't dividing, how do they know those cells aren't just dying? Or, maybe they're just temporarily stunned by the trauma of a literal blade slicing through their neighborhood.
Well, that's exactly the skepticism the scientific community brings to a claim like this. You can't just point to a stationary cell and declare it senescent. You have to prove it, both functionally and structurally.
Right. You need receipts.
Exactly. And to get those receipts, the authors of this paper adhered to a really rigorous set of standards called the MICEC guidelines.
ICSE.
Yeah, MICSE. It stands for Minimal Information on Cellular Senescence Experimentation.
Okay, that is quite a mouthful.
It is, but it's crucial. According to ICE, a genuinely senescent cell, a bona fide zombie cell, must be in a state of stable cell cycle arrest, and it has to exhibit permanent major changes in 3 specific areas.
Okay, what are they?
First, it's transcriptome, which is the RNA it produces. Second, it's proteome, which are the physical proteins it builds. And third, it's secretome, the molecules it actively secretes into its environment.
So it's not enough for the cell to just hit pause. It has to fundamentally change its entire identity and behavior. It's like identifying a suspect, right? You don't just want a footprint. You want a fingerprint, an eyewitness, and DNA.
That's a great way to put it. You need multiple lines of evidence. And they found 5 highly specific markers, 5 fingerprints, right at the edge of the wound.
Okay, break those down for us. What's the first one?
First, they looked at the proteome and found a massive upregulation of a protein called P21.
P21?
Right. It's encoded by the TTTK1A gene. And its primary job is to act as the master stop switch for the cell cycle. When a cell is flooded with P21, it physically cannot divide. It is locked down.
Okay, so P21 slams the brakes. But just because a car has the brakes applied doesn't mean the engine is off, right?
Sure.
Like, the cell could still be trying to copy its DNA in the background. How did they prove the actual division process was truly arrested?
By looking for the absence of 2 specific proliferation markers, Ki67 and EDU.
Okay, what do those do?
Well, Ki67 is a protein that is strictly present when a cell is actively dividing. If you don't see it, the cell isn't proliferating, period.
Got it. And then the other one, EDU.
EDU is even more definitive. It is a thymidine analog, basically a synthetic building block of DNA. In these experiments, researchers introduce EDU into the animal system.
Okay.
And if any cell is actively unzipping and copying its DNA to divide, it will eagerly pull that EDU into its newly built DNA strands.
Ah, I see. And when they looked at the P21 positive cells at the wound edge, I'm guessing they found none of that synthetic building block?
Exactly. Almost 0 Ki67 and 0 EDU incorporation. The engine wasn't running. The cells were completely frozen.
Wow. But
to satisfy the AMICE guidelines, you also need structural changes. You need to see the cell physically alter. That brings us to the third marker, which is a dramatic reduction in a protein called lamin B1, or LMN B1.
Lamin B1, what does that do?
It forms the physical scaffolding of the cell's nucleus, the vault where the DNA is kept. In typical aging, senescent cells lose this scaffolding. The nucleus actually changes shape and starts to degrade.
It just collapses.
Yeah, essentially. And the researchers saw this exact same nuclear degradation in the wound cells at just 90 minutes.
That is wild. Okay, so the cell is frozen, it's not copying DNA, and its internal scaffolding is collapsing. What else? What's the fourth marker?
Fourth, they observed significant hypertrophy. The cells physically swelled up, drastically increasing their soma or their body size.
Wait, why swell up, though? If they are completely stopped, shouldn't they just sit there, like, dormantly?
You would think so, but that is a key characteristic of senescence. The cell is physically arrested, so it can't divide, but it is still highly metabolically active.
Oh, I see.
Yeah, it's churning out proteins and molecules. But because it cannot divide into two cells to relieve that internal buildup, the pressure and volume just increase, so it swells up dramatically.
That makes total sense. Using our earlier analogy, it's like the factory is still running, but the shipping doors are locked. Nothing is leaving the warehouse, so the building just has to expand.
Precisely. And that metabolically active state leads to the fifth and final marker. This one involves a really fascinating process called ALISE.
ALISE, like the name?
Yeah, ALISE. It stands for accumulation of lipids in senescence. The researchers observed that these cells started hoarding lipid droplets, tiny vesicles of fat in places they normally shouldn't be.
Huh. How do they even track that?
They track it using a protein marker called PLN2, or paralipin-2. It acts like a coat around the surface of these fat droplets. Now, in healthy, uninjured skin, PLN2 is almost exclusively found in the sebaceous glands.
The oil glands.
Right, the oil-producing glands, but in the wound.
Let me guess. It's everywhere.
Within that 90-minute window, PLN2 completely flooded the injury site. And it perfectly overlapped with the exact same cells expressing that P21 stop protein. These cells were rapidly accumulating ectopic fat.
Okay, this is a lot to take in. We'll do a quick recap for everyone listening, just to lock this in. Time for our first little bullet point summary.
Go for it.
Okay, so point 1. Senescence means a cell completely stops dividing. Point 2. This usually takes weeks, but in a fresh wound, it takes exactly 90 minutes. And point 3. We know they are genuinely senescent because the researchers checked every single box.
The 5 markers.
Right. They slammed on the brakes with P21. They stopped tolling in building blocks like ADU. Their nuclear scaffolding, lamin B1, collapsed. They physically swelled up into giant warehouses. And they started hoarding lipid droplets with ALISE. That is an airtight case.
It really is.
Which makes you wonder. Think about a time you scraped your knee. Could you have ever imagined that within an hour and a half, the cells at the very edge of that scrape were deliberately transforming into swollen, fat-hoarding, non-dividing zombies?
It profoundly shifts our understanding of how dynamic the skin really is. But it also presents a massive mechanical paradox for the researchers to solve.
Right, because here's where it gets really interesting. The timing still makes absolutely no sense.
Exactly.
If making a cell senescent requires fundamentally changing its proteins, like flooding it with P21, how does a cell physically achieve that in 90 minutes? Because normal cellular biology has a speed limit, right?
It does, a very strict one.
Usually a cell has to realize there's a problem, send a chemical signal to the nucleus, unzip the DNA, transcribe that DNA sequence into messenger RNA, export that mRNA out of the nucleus, and then finally use ribosomes to translate it into a physical protein. You can't do all of that from scratch in an hour and a half.
You are absolutely correct. The traditional path transcription followed by translation is just way too slow for a 90-minute response.
So how are they cheating the system?
The answer lies in a mechanism the authors identified as transcription-independent translation.
Transcription-independent translation. Okay, walk us through that.
They made a startling discovery. Your homeostatic skin, meaning your normal, healthy, completely uninjured skin right now, maintains an unusually high basal level of pre-existing CDKN1A mRNA transcripts.
Oh, wow. So it's already there.
Exactly.
It's like keeping a fully loaded fire extinguisher on the wall. You don't wait for a fire to start and then draft blueprints to manufacture an extinguisher from scratch. By the time the factory builds it, the building has already burned down.
Right.
Instead, your skin cells are using this pre-existing mRNA as a biological fire extinguisher. It's just hanging there, fully loaded, ready to deploy instantly.
That is a perfect analogy. The mRNA for the P21 protein is fully synthesized and just sitting in the cells waiting.
But wait, if the mRNA is just sitting there all the time, fully loaded, why isn't the cell always senescent? Why doesn't the extinguisher accidentally go off and just constantly age our healthy skin?
Because the pin is firmly in place. Under normal conditions, that preloaded mRNA is tightly bound by specific inhibitory proteins. These proteins act as molecular bouncers. They physically restrict the mRNA from being exported out of the nucleus. Since it can't reach the ribosomes in the cytoplasm, it can't be translated into the P21 protein.
Okay. So the bouncers keep it locked in the VIP room?
Exactly. However, the moment an injury occurs, the sudden, drastic change in the cellular environment causes these inhibitory proteins to just fall off.
So the physical trauma of the injury itself pulls the pin.
Yes.
The mRNA is released from the nucleus, hits the ribosomes, and instantly churns out the P21 protein. That skips the entire slow transcription phase entirely.
It skips it completely. That's the secret to the 90-minute speedrun.
That is brilliant. Did the researchers figure out exactly what chemical signal triggers those inhibitors to drop?
They did. They proved that this rapid-onset senescence is driven by mTOR signaling.
mTOR.
Yes, lowercase m, capital T-O-R. It stands for the mechanistic target of rapamycin. It's a major metabolic signaling pathway in cells that regulates growth and survival.
Okay, so how did they prove mTOR was the trigger?
The researchers tested this by applying two well-known mTOR inhibitors. These are drugs called everolimus and rapamycin. And when they suppress the mTOR pathway with these drugs, the rapid appearance of P21 positive cells at the wound edge was significantly reduced.
Wow. So they blocked the signal and the extinguisher never went off. Did they check if this was just a standard reaction to DNA being damaged by the cut? Like the scissors slice the DNA and the cell panics?
They explicitly ruled that out, actually.
Yeah.
Usually, severe damage to DNA triggers something called the DDR, or DNA damage response, which does eventually lead to senescence. But the researchers found this rapid wound response operates completely independently of the standard DDR.
Oh, interesting.
Yeah, it is an entirely distinct, pre-programmed, mTOR-driven release of stored emergency instructions.
Okay, it's time for another mini recap for everyone listening. Let's bullet point this mechanism.
Let's do it.
Okay, point 1. Skin cells stockpile that CDKN1A mRNA in advance. Point 2. The physical injury unlocks this mRNA from the nucleus, dropping the bouncers. Point 3. The cell immediately translates it into the P21 stop protein, completely skipping transcription. And point 4. This whole process is driven by mTOR signaling, not standard DNA damage.
Spot on.
Which brings up a really fascinating thought for you listening. If your skin is essentially preloading an emergency response system, keeping it locked and loaded 24/7, what does that tell you about how evolution views the physical barrier of your skin? It treats a minor scratch not as a mild inconvenience, but as an absolute existential emergency.
Because it is. The skin is our primary barrier against pathogens.
Yeah.
Any breach is a life-threatening vulnerability in the wild.
Right.
But this brings us to the core functional question.
Right. Why do it at all?
Exactly. We have established that these frontline cells arrest their division immediately. But if we connect this to the bigger picture of wound healing, why do that? If a wound needs to close to stop infection and blood loss, wouldn't you want every single available cell multiplying as rapidly as possible to fill the physical gap?
It feels incredibly counterintuitive. You have a literal hole in your defense, and the cells directly at the edge are instructed to just refuse to multiply. What is their job if not to divide and rebuild the tissue?
Their job is to act as the command center for the rescue operation. This is where we need to introduce the concept of the SASP.
SASP.
Yes. It stands for senescence-associated secretory phenotype. Remember earlier when we said senescent cells change their secretome, the molecules they release?
Yes, the third part of the MICSC guideline.
Right. Well, while these P21 positive cells are physically arrested and not dividing, they are incredibly active chemically. They begin aggressively secreting a highly specific cocktail of pro-migratory and pro-inflammatory factors into the surrounding tissue.
Wait, so they sacrifice their own ability to divide so they can chemically direct the tissue repair?
Yes.
It's like they become traffic cops or like site foremen on a construction site. They sit stationary at the very edge of the wound, pumping out this SASP cocktail to command the healthy non-senescent cells behind them to migrate forward and physically bridge the gap.
Traffic cops captures it perfectly. They are orchestrating a process called re-epithelialization, which is just the scientific term for the resurfacing of the wound.
Re-epithelialization. Got it.
Now, To prove that these stationary cells were actually directing the healing, the researchers designed experiments using both mouse and porcine models.
Porcine, so pigs.
Yes. Porcine skin is remarkably similar structurally and immunologically to human skin, making it a gold standard for wound healing research.
Makes sense.
So the researchers basically asked, what happens if we forcibly remove these traffic cops from the intersection?
How do you even do that? How do you selectively target just the rapid-onset senescent cells without damaging all the rest of the healthy skin?
They used two distinct approaches to be absolutely certain of their findings. First, they used a pharmacological approach.
So drugs.
Right. They treated the wounds with a specific drug called UC2288.
UC2288?
Yes. This drug attenuates or weakens the CDKN1A mRNA. That significantly reduces the amount of P21 protein the cells can produce. This effectively prevents the cells from ever entering that rapid senescent state.
Okay, so the drug stops the traffic cops from ever showing up for duty. And what was the second approach?
The second was genetic. They utilized genetically modified animals known as P21 ATAC mice.
P21 ATAC, that sounds intense.
It is. These mice are engineered with a brilliant biological kill switch.
A kill switch.
Yeah. Researchers can administer a specific, otherwise harmless chemical to the mouse, and it will trigger targeted apoptosis programmed cell death exclusively in cells that are actively expressing the P21 protein.
Wow. That is incredibly precise. So in one model, with the drug UC2288, they chemically prevent the cells from becoming senescent. And in the genetic model, the P21 ATAC mice, they basically execute the senescent cells the moment they appear.
Exactly.
So if the traffic cops are gone, does the whole intersection jam up? What happened to the wound?
The healing timeline completely collapsed.
Really?
Yeah. The researchers found that if you eliminate or suppress these senescent cells during the early phase of healing, right after the injury wound closure is drastically delayed.
Because the healthy cells don't know what to do.
Exactly. The healthy cells deeper in the tissue lack the chemical signals they need to migrate properly. Without that SAP cocktail guiding them, the whole repair mechanism just stalls out.
Did it matter when they removed the senescent cells? Like, what if they let them do their job for a few days and then cleared them out?
That's the crucial nuance in the data. If they applied the chemical inhibitors or triggered the genetic kill switch later in the healing process, after those initial critical days, it had no negative effect on wound closure at all.
Oh, wow. So they are strictly an initial emergency response requirement.
Exactly.
Okay, let's do our third mini recap for everyone tracking this at home. Point 1. Senescent cells aren't dead. They are highly active and secrete SASP factors. Point 2. These factors act like traffic cops, causing other healthy cells to migrate and close the wound. And point 3, if you block P21 early on, either with the drug UC2288 or the genetic kill switch, you ruin the healing timeline.
Perfect summary.
It just completely flips the script, doesn't it? Have you ever considered that the biological state we usually associate entirely with decay and aging and wrinkles is actually a fundamental requirement for regeneration and repair?
It is a stunning paradox.
Yeah.
But it also introduces a very serious long-term physiological problem.
Oh, right, because decades of aging research have taught us that lingering senescent cells are toxic.
Very toxic. If they stick around for months or years, that exact same SESP cocktail causes chronic inflammation. It degrades surrounding healthy tissue, and it drives age-related diseases like osteoarthritis and even cancer.
Right. An emergency alarm is only useful if you can eventually turn it off. If the skin gap is successfully closed, the traffic cops need to go home. So what does this all mean for the cleanup? How does the body prevent these hyperactive inflammatory cells from turning against us once the wound is physically healed?
It executes a flawless, highly regulated disappearing act. The researchers meticulously tracked the population of these P21 positive cells over time.
Okay, what was the timeline?
They found that the number of senescent cells at the wound site peaks exactly at day 3 post-injury. That is when they were driving the absolute peak of the cellular migration and repair process.
Day 3, okay.
But by day 28, which is roughly 2 weeks after the wound has physically closed over and resurfaced, they are entirely gone.
Entirely.
Entirely. When the researchers looked at unwounded skin and fully healed skin at day 28, the count of P21 positive cells was literally 0.
0. They don't just, like, reduce in number or go dormant. They completely vanish. Where do they go? I mean, they can't just revert back to being normal, healthy cells, can they?
No, they can't. Senescence is a terminal state. There is no going back.
So how are they cleared out?
The clearance mechanism actually depends on exactly where the cell is located within the skin's layers. In the upper layer, The epidermis, the senescent cells, specifically the keratinocytes, are simply pushed upward as the skin repairs itself from below.
Oh, I see.
Yeah. They're ultimately removed via shedding. They literally just physically flake off as dead skin.
That handles the surface, but what about the cells deeper down, the ones that can't be shed?
For the senescent cells deeper in the dermis, like fibroblasts, they obviously cannot be shed. So instead, they are eliminated through apoptosis, that programmed cell death we mentioned earlier with the kill twitch mice.
Right.
But naturally this time. Unlike necrosis, where a damaged cell violently bursts and causes massive inflammation.
Which we definitely don't want.
Exactly. Apoptosis is a quiet, highly organized dismantling of the cell from the inside out. Once the wound is closed, the local microenvironment changes, the survival signals vanish, and these deep senescent cells initiate their own destruction. Their remnants are then quietly cleared away by the immune system's scavenging cells.
That is incredibly efficient. It's like a pop-up emergency response center that gets completely demolished and swept away the moment the crisis is averted, leaving absolutely no trace behind.
And this highlights a massive physiological contrast that the researchers point out. This transient, rapid-onset senescence is vastly different from what we see in traditional chronological aging.
How so?
Well, when senescence is caused by the slow, gradual accumulation of DNA damage over decades, or by massive systemic hits of UV radiation or chemotherapy, the body's immune system often fails to clear the senescent cells. They accumulate in the tissues. And that accumulation is what causes the pathology and decay of aging.
So the problem with aging isn't the senescence itself, it's the failure of the cleanup crew. All right, time for our final mini recap, final bullet points for you listening. Point 1, rapid senescence is temporary. Point 2, it peaks at day 3 and is completely gone by day 28. Point 3, the body clears these cells out efficiently through shedding at the surface and programs cell death deep in the tissue.
It's a completely different beast than the cellular aging that causes our bodies to break down over time.
It really is. So if we connect this to the bigger picture, what are we walking away with today?
The findings in this paper force us to redefine a core biological concept. Cellular senescence is not merely a disease of aging or a failure of cellular maintenance. In the context of the skin, it is a highly evolved, rapid, transcription-independent survival mechanism. It is a vital adaptation preloaded into our biology, designed to ensure we survive the daily physical traumas of interacting with the world.
Today, we discovered that your skin isn't just reacting to damage. It is actively anticipating it. It maintains a secret stockpile of pre-synthesized mRNA, a molecular fire extinguisher locked inside the nucleus, just waiting for the physical trauma of a simple paper cut to pull the pin and deploy a protective aging mechanism in 90 minutes flat.
It's an incredibly complex, elegant system.
Happening right under your nose. Or, rather, right on your finger. Which raises a deeply fascinating question for you to explore on your own as we wrap up today's deep dive. If the skin has evolved this incredibly complex, preloaded emergency brake to survive physical trauma, what other vital organs in your body might be hiding a secret reserve of pre-existing transcripts, just waiting for the exact right trigger to instantly transform their biology? Until next time, keep diving deep.